Hello, everyone, and welcome to the Relapse Refractory Myeloma Patients chapter. My name is Audrey Burton-Bethke, and I'm the Myeloma Community Manager. I want to start by sharing a couple slides with you just by way of announcement. So let's get that up and running. All right, so today's session will be recorded and it will be sent out to you within 48 hours of the presentation concluding. I'm grateful for your support and your participation here today on a Friday morning or evening, depending on where you are in this world. We're going to be hearing from Arjun Van Manen. I Americanized his name, who I will introduce formally in just a moment. As always, a special thanks to our sponsors, Bristol Myers Squibb, Amgen Oncology, Karyopharm Therapeutics, AbbVie, Adaptive Biotechnologies, Genentech, Oncopeptides, and Takeda Oncology. So today's topic is going to be utilizing precision diagnostics to identify high-risk myeloma at diagnosis and relapse. Knowing what type of myeloma that you have allows you to better fight it. Something you might not know is that you can relapse with a different genetic type of myeloma than you had before your remission, possibly a genetic high risk factor. In addition to this, researchers are working extremely hard not only to detect high risk genetic factors in the beginning of diagnosis or re-diagnosis, they're also exploring the use of precision medicine to discover what treatments are best for different types of multiple myeloma. One of the biotechnological companies doing this type of research is Diagnostics or SkylineDx, and that is why we wanted to invite one of the representatives here today to speak to our group. So it's now my pleasure to introduce our keynote speaker to you. Arjun Van Manen is the EVP of Business and Commercial Development at SkylineDx. He is a strong commercial professional with a track record in clinical research, international marketing strategy, market access, commercial excellence programs, and business developments. His primary fields of expertise are oncology, hematology, and surgery. He has also had experience in pharmaceutical, medical device, and diagnostic fields. Prior to working at SkylineDx, he worked with organizations such as Bristol Myers Squibb and Takeda Pharmaceuticals. He is fluent in Dutch, German, and English. So Arjun, the time is now yours.
Thanks, Audrey. Thanks very much for the introduction and for the invitation, of course, to be able to share this with your group. Audrey, if you can confirm that you can see my presentation, that would be great.
Yes, I can.
Perfect. So, well, welcome to.
There's like a buzz in the background. It wasn't there before, so I'm not sure what that is.
Didn't change anything. I can. That makes a difference.
Oh, it's okay. You can just keep talking. It's okay.
Okay. Well, if not, I'll try to dial in by phone and do something different. So please let me know if I need to. So thanks again for giving us this opportunity to share the research that we did and how that relates to the efforts to provide some kind of direction towards personalized medicine and personalized diagnostics. So this might be a repetition for most of you, but when we look at the facts, you see that multiple myeloma is a relatively rare cancer, and it's often more prominent in the age category above 65. So we see that the incidence is higher above 65, and that the median age, about 50% of the patients will be below 69, and 50% will be above the age category. It represents about 50% of the hematological diseases. And when we specifically zoom into the US, it will be about 32,000 patients being diagnosed with the disease every year. Well, the interesting thing is, although it's a rare cancer, it's considered one of the very complex and heterogeneous cancers. And to the remark of Jenny in the video, still regarded as incurable. So what you would see is that over time, the the treatment would give you a plateau remission, so you would do well under treatment, the disease will be under control. But at a moment in time, it might be that the disease will relapse, come back, and then the treatment needs to be started again. And then it would go into a different concept of how far the plateau can be reached and how long that would be. So all the treatments that are currently being developed or given or have been given are all about achieving that depth of response, to maximize that depth of response, but also strive for the most durable response when it comes to the individual treatments. Although there has been a tremendous progress over time, and there's a huge progress when it comes to the different treatments that have been introduced to the market, you still see that there's data coming out or has been published that it is really difficult to predict how many lines of treatment or how many relapses a patient might be confronted with. The other thing that is important to realize is that it really goes into what Audrey says, you have that clonal expansion. So it starts at some point as something that is not necessarily defined as a multiple myeloma yet. It can be a precursor, it can be something that they call MGUS, then something that could be defined as early melanoma or myeloma, sorry, and then over time you would see the different stages of severity play in. But most often, the patients will arrive at the doctor very symptomatic. So the consequences, the symptoms associated with the disease are very prominent and are very much impacting the quality of life and the overall comfort or day-to-day living that the patient has. These might be really well already known to you, but the CRAB features are one of those aspects that are then measured to then see what the severity of the disease is, correlated to then the percentage of the plasma cells. So the multiple myeloma lives in the plasma cells in the bone marrow, and then the number of plasma cells or the percentage of plasma cells would give you the tumor load information, and then that's correlated to what you would see here. So the calcium levels, so often the patient can come in with bone lesions because the calcium levels have been affected, renal failure, anemia, and then the other features that have been all recorded into guidelines when it comes to the official diagnosis. So when the diagnosis is made, you see that different tests are being performed. So the predominant one would be a blood test. There are tests being done or performed on the urine. And what our test really focuses on is the bone marrow. So our test really focuses on a depth of analysis that is done on the aspiration of the bone marrow itself. The bone marrow is often done in most patients for several reasons. And one of the dominant ones is that they really want to look at those plasma cells. So what is the percentage? How many cells are there? They have different methodologies to investigate those, but they also want to look to the morphology. So the morphology is really the form, the shape, the size of the cell, and then get distinct features out of that. Something that's relatively new is then genetic profiling, and that's where we developed our tests for. Most often in current practice, you would have the flow cytometry done and the FISH done. And more is learned is that the genomics are actually really important to better understand, because what they see is that the genomics have different abnormalities when it comes to the disease stages. So they have some kind of evolution when it comes to the different phases of the disease. You see something called clonal tiding. I'll show you that in the next slide. And it's related to mutations that might be acquired, translocations that might change over time, or abnormalities that might occur that were not there before. A principle that makes it even more complex is clonal tiding. So you see here a distribution of different clones that were present at diagnosis. A patient would get the treatment in this example from this publication with lenalidomide and dexamethasone. You see that it has effect. The patient goes into remission, but then there's a relapse. And then all of a sudden, a clone that was less prominently there then has a dominant position at that first relapse. So this process is really looking at the almost evolution over time within different phases of the treatment that could be correlated to different aspects. But this also makes it really imperative that the physician needs to look at the optimum treatment strategy almost already at the onset of the diagnosis, anticipating that there will be several remissions where you might need to do different principles of treatment. So when we look at that clinical landscape, and that has been one of the most exciting things I would say for multiple myeloma, is that there has been an influx of different treatments, and not just on the treatment itself as a standalone, but also in the combination of the different treatments that are possible, the sequence in which they are given, which on one hand is a very positive development because it just gives a lot more of opportunities. On the other hand, it also creates a level of complexity. What should be the first treatment, the second treatment? And to the video, I would definitely say that there's a need to really have a clear and good understanding between the physician and the patient on what the potential impact or consequence or toxicity profile might be of these treatments. So what we also see then in the clinical domain, in the guidelines, and this is worldwide, so if it's US or Europe, is that almost every year new data comes out, which makes it even more complex to keep track. But it also speaks to the fact that the initiatives that are then facilitated by Myeloma Crowd provide you that opportunity to stay educated and to really see if there is something new on the horizon or something new being introduced that provides a potentially different aspect to treatment. The most common regimes, I would say, is that first there would be an induction therapy. It could be a discussion about when then the optimal combination will be, if that's two treatments combined, so a doublet or triplet. You would see that especially in Europe, then an autologous stem cell transplantation will be standard for the ones that are considered eligible. In Europe, that's often age dependent. What we see in the US is this is really also related to the hospital, the clinical practice of the specific physician, and how they believe that one would give or provide better results in comparison to another. Then you get a consolidation treatment, which is short course, and then the maintenance to make sure that the results are durable. The field of multiple myeloma, so not only the disease itself, but also the clinical and therapeutic landscape is incredibly complex, as the area of multiple myeloma, as I think the area of pharmaceutical introductions that saw the most pharmaceutical introductions in the last 10 to 15 years. So although it being a rare cancer, you just see a lot of things being introduced. And what we know is that there's still a lot to come as well. So CAR T cells is a relatively new treatment, your BCMA, there are a lot of different opportunities. And then the question plays into the discussion, when should you give what treatment and what kind of consideration would you have? Well, risk stratification is one of those elements that would give you a potential guidance in what the optimal treatment path would be. So at this moment, this is a slide deliberately took from a conference. So you see a lot of the discussions between the key opinion leaders, the medical professionals, because they all strive for the goal of personalized medicine. So they really realize that multiple myeloma is more a disease of the individual instead of a group of people. So the question that they really have is, when we come to personalized medicine, how feasible is that and how can we do that? Well, there are a couple of principles that they need to take into account. The first one be to maximize that outcome. And the maximization of the outcome is really, how can you realize the best depth of response, meaning that you have the least amount of myeloma cells on the basis of that treatment. But what you also want to do is have a durable response. So you want to have that effect not for short term, but for a longer term. It also needs to be acceptable to the patients, meaning that it has to have toxicity that a patient will be able to endure and doesn't impact the quality of life in the risk-benefit of having a choice for a specific treatment. And the other aspect, and this is one of the important ones, is that at some point, if the relapse comes, does it mean that that patient has become resistant or the disease has overcome the potential benefit of that treatment? Therefore, a new treatment as a new mechanism of action, because they would address a different clone, or they would be more synergistic with another combination, would be given a different prospect in that treatment line. The focus has been really on predicting patient outcomes and targeted treatments. What SkylineDx did is, from the research that we always did with clinicians, research group, and academic institutions, is to focus on, at first instance, the prediction of the patient outcome. So what we saw, and these are all publications on different conferences as well, is that the medical society is still learning a lot on how to define high risk. So there are different definitions currently out there for high risk. There are different schools of thought. There are some common denominators. So there are a lot of publications out there that said that if there are certain genetic abnormalities, or if you have the elevated serum beta-2 microglobulin, that will be correlated to reduced renal function, therefore less favorable prognosis versus if you would not have that. A recent school of thought would be, if you have a single abnormality, that would give you some information, but you get a worse message if you have 2 or 3, because that means that the disease will progress really rapidly, or you would fall back into a relapse relatively soon after the initial diagnosis. So a concept that is relatively new is gene expression profiling. Our company, Sky for Skyline, just the abbreviation of the company, and the 92 genes that are then analyzed. I will give you more understanding in the presentation, but that's what we've developed for the last almost 7 years to really see, can we standardize and provide that accurate understanding at the onset of the disease, so really at the point of diagnosis, but also at the point of relapse and refractory. The main message should also be, so although all these elements are recognized by all the major guidelines, if it's the American groups, if it's the international groups, or if it's European groups, they all realize that is not a single feature that you should take into account. It's not just your revised ISS or your ISS. It's not just your FISH markers. You should take it as a holistic point of view and take all these markers into account, because it can also be related to the performance status that you might have, or the age that you are, to give a different prospect on how you would define risk and how you would define the likelihood of progression. One of the things that I think might be underestimated is that there might be a very strong personal preference from the patients or the caregivers when it comes to a given treatment. So if the clarity is there and the mutual understanding is there on what then the characteristics are of the disease and what the preferences are of the patient, in mutual understanding with that physician, there might be more clarity on what then the optimal treatment path would be. And that's also a form of personalized medicine. So Sky92, the discovery. So the background is that one of the clinical trials that has been published now quite some time ago in 2012, it's the HOVON 65. It was performed in the Netherlands and in Germany. They collected a lot of information on these patients that were included in the trial. So the platform that was used is microarray. Microarray is a platform that would take the bone marrow, it goes through a process, you focus on the myeloma cells, and then you would be able to read the gene expression. And the gene expression is then on the microarray, which is almost like a chip, and then it would give you the information which of the genes are switched on or off. That's kind of then the expression levels that you would have. So with all that information, the bioinformatics, so it's a computational biology group, together with the clinicians said, could we correlate that to some sort of conclusion? So does it give us an understanding of a pattern or a trend or a subgroup of patients that you can signify and get to a certain conclusion? So what they found is that there were 92 genes that if they would be switched on or switched off in a certain combination, but if they would be there, they would be highly significant for demonstrating that that patient would progress much faster than the rest of the group. And it was about 20%. So that was the starting point of our research. So we said that's a really important finding. We really need to see if we can validate that in other groups as well to really make sure that this is not a single discovery that we do, but that it definitely gives additional information across different geographies and across different patient cohorts. So as a company, we want to bring this also to clinical practice. So we go through the different steps, almost like you would have with pharmaceuticals. So you go through phases of research before it's actually then admitted to the market. And like it is with a pharmaceutical, you then have to file a submission to get marketing authorized to see that you're safe before you go into the market. So the last 7 years, we did these phases of the research and prepare for that market launch. One of the key aspects that we then we had in the research was there's already so much out there. There's a lot of information when it comes to the mutations, what you saw before on the presentation when it comes to the either it's double hit or single hit, but a lot of molecular information is already collected from the patient and from the disease. So one of the question was, would one replace the other? And there's still data coming out there. This is a relatively recent publication from the Royal Marsden in London, the group. They said that gene expression profiling and genetic risk markers actually look at something different. They might have overlapping biological properties, but in the end, they give you a better understanding as a whole when you combine that information. So you increase your prognostic sensitivity and you characterize the disease better on a molecular level. So for us, one of the important things, especially from the scientific point of view, is now that you have the 92 genes, what does it give you on the biology? Do we then understand the disease better? Is there any kind of group in there that might be more indicative for a certain treatment? Does it tell you anything about how cells divide and multiply? I would love to say that we have the answer to that, but this is really still early days and this is still ongoing. So we see that there's a lot of information already there that certain genes, when they're switched on or they're switched off or they're in the signature, would give you information on cell growth and proliferation. They would give you indication on DNA repair, but we have not found a single principle yet on the basis of the signature that could be correlated to a specific treatment. So the workflow is standardized. So this is what you really need to do if you would come to an analysis. So the Sky92 is performed on the bone marrow. So the bone marrow is aspirated. The specimen would go through a quality standard procedure. We would need a certain amount. So it says 2 to 10 milliliters of bone marrow, because this is really dependent on the quality. Then we need to have the RNA, which is then the source for which we can do the plasma cell purification. I don't want to make it too complex, but it goes through really diligent steps in the lab with quality control measures to really see that we come to the purest group of cells that we can analyze to really understand what then the characteristics are of the disease. So when we have the optimization, which is mostly analytical validation. So the first step is, can we do this? Can we make sure that we have a standardized procedure? Do we get repeatable results? Would it be different if we would do it in the lab in San Diego, or if we do it in the lab in Rotterdam, or 2 locations, which has been all proven and published that that's exactly the same. So we get really good results. We also want to see, so how does it affect when you get a different treatment? So if you are predicting the prognosis, is that not necessarily correlated then to the treatment that was in that investigational group or that was in that specific study. So what our group did, we did it 16 times. Don't ask me why it is 16 times, but our group worked together with different research groups, academic institutions from the US and from Europe to take different trials, mostly trials, but also real-world patient data. And then there should be banked bone marrow samples, then reanalyze those, and then correlate that to the outcome. Then you go back to the data because it's all retrospective, and then you would have the same question. Will we get still the information on the prognosis of these patients as we had in the discovery cohort? And what we saw across the board, if you're relapsed refractory or if you're newly diagnosed, it's almost about the same fraction of patients. So if you're positive on a SkylineDx signature, if you have that SkylineDx signature presence, that means that you are a fast progressor and you really have an aggressive form of the disease that could be defined as high risk, but it means that you don't respond well to any of the given treatments that were investigated in these cohorts. I would say that in most literature, that's exactly what you would also see for some of the other markers or some of the other information. So the majority of the treatments that have been developed give a tremendous progress and a really positive results for standard risk patients. There's still a high unmet need to make fast progress, fast progress. So a positive change for patients that can be considered high risk. Here again, what you really want to understand is what then the comparison is. So if you use one compared to the other. So SkylineDx is in this research then compared to all the different features that were currently in clinical practice or at the time of clinical practice, because it was 2015. And what you see that if you would use the SkylineDx alone, or if you would combine it then with something that's readily used in the clinical practice ISS, you get the best understanding of the aggressiveness of the disease. And this is for overall survival. The other thing that we found interesting is that if you look at the SkylineDx, is it there or not? SkylineDx, if it's there, you know it's not good. So it really gives you an understanding that you have a bad variant of the disease because it's fast progressive or fast progressing. But if you don't have it, what does it give you for information? So this is also ongoing research that we did, but if you're in the good group, which is still about 80% then of the patients, taking into account that this is mostly newly diagnosed, if you were to correlate that with your ISS and you have the positive ISS, meaning the ISS1, you're actually in a good group and you consider that low risk. Low risk is a relatively new concept in multiple myeloma, but there is a group, and in this case, it's about 30% of these investigational groups that do really well over a longer period of time. So although this is, I would say, in some of the earlier phases of research, this is also a very different conversation that you could have with your physician. So if you're in the good group, would you wait for a more aggressive approach, or would you like to still have the fast aggressive approach because you might get the depth of response that you're looking for to achieve? So there are different considerations there, but what you do see, this is a Kaplan-Meier, meaning this is a graph for how fast the progression is. You see that there's a big difference between the green group and the red group for the top line and the bottom line. So again, the physicians and the researchers are redoing some of the research or confirming some of the research when they say now that there is a new definition of high risk, meaning if you now would look at 2 cytogenetic markers or aberrations and you would compare it to SkylineDx high risk, would that then outperform genetic expression or gene expression profiling? Well, this one is from last year, and this is again a UK group, but what you see is that you find an additional 10% that are high risk and they're not found by any of the other markers. Short delay. So what you see here is as well is this is a very recent abstract. So all of the research that we do as a company, we submit it to, well, at first instance, we submit it to a conference or we submit it to a congress. Often it will be in the US ASH, or in this case, it was the European Myeloma Network conference, or it's EHA, which is then the European Hematology Association in Europe. Just because we want to share everything that we do, and we do not do it alone. We often do it with the clinicians and the lead investigators. But here what you see is that if you were to take the definition of high risk by having a translocation for 14 or a deletion 17p, should actually maybe do it on the basis of 1D, that's more illustrative. So here you look at the high risk group. So these are the patients, the pink line, the bottom line, that's the group of patients that have the bad variant. So they have the presence of the SkylineDx signature. So if I now would have defined that group differently, I would have taken the position of a patient is high risk when they have 414 and or deletion 17p, or translocations. Would they then all be in that pink group, or would they be somewhere else? Well, what you see is that those two lines are almost overlapping. So that means that, I'm not sure if you can completely read it at the bottom, but there are almost as many patients in the bad group that have been tested positive for those translocations as negative. So if you would relate that to clinical practice, so you do your workup, you get all the information, hopefully everything's shared with you, you have the conversation with the doctor, and the doctor would say, you actually have a really positive outlook of your disease. You do not have any translocations there that we feel are indicative of a bad variant of the disease. Therefore, we consider this kind of treatment, and they want to discuss this with you. But what you see here is that that's not necessarily the case. It could just well be that you are negative when it comes to the presence of these, but you still might have a fast progressive variant because there's not the complete understanding of all the aspects of the disease. So we try to capture that a little bit in this, and it is for newly diagnosed, but we try to capture that a little bit in this decision tree. So when you would have the most accurate understanding of the risk, when it comes to the onset of the disease, so when you get the initial diagnosis, and you would apply that low risk principle, you could actually have different categories. And I really appreciate what was said on the video at the beginning. If you're educated enough and you know that you're in the high risk group, you might want to find out if there is a specific clinical trial or a novel treatment that you can have access to. It could even be a case where you then said that you would consider supportive care instead of going into a trial setting. But it is all about understanding and being informed so that you can make the right decisions together with the physician. One of the exciting things I would say that has been introduced in the last couple of years is minimal residual disease monitoring. So there are now tools, especially in the US, that look at the disease levels or what's remaining of the disease, so that you can actually look at the durability and then see if you can vary the disease path by maybe giving less or more escalating treatment or de-escalating treatment if the desired effect has been achieved. So the clinical utility, so where we are now in the US specifically is, so we focused on going back to the phases of research. So we focused first on standardizing the process, doing the analytical validation, then doing the clinical validation, meaning does it do the same when we look at a different group of patients, is that we then strive for getting the evidence there, which is clinical utility, meaning that we can develop this, and in theory, it sounds really good. And if we would ask our colleagues, they're really proud of all the research that they did. But does it also translate then to the clinical practice when it comes to the physicians that might use it in clinical practice? So a couple of years ago, we started a US trial, which is the Promise trial. You can also find it online if you want to have more information. But the trial itself has run in these institutions, where the primary objective was really to look at if we do not have this information, what would we advise for treatment? If we now add the information of Sky92, would that then change the first consideration of treatment that doctor, predominantly the doctor, might have to discuss with the patient? The researchers that participated in this study actually, I wouldn't say surprised themselves, but at least they came to the conclusion that this is a very recent publication, that they found that it really renewed their view on how you do a clinical risk assessment. They found that it could give guidance on how you can optimize your treatment plan, but you can also adapt it over time if you would incorporate that minimal residual disease risk status. And then they actually did it in both directions. So we anticipated that it might be more dominant for most physicians, that if they consider someone to be, or if they consider the disease to be standard risk, and then they were informed that a patient will be high risk, to escalate the treatment. Just to be on the safe side, you would give the most effective form, because you really want to have the best effect, so that you also are prolonging the period of remission. But what we also saw is that there were quite some physicians that, when they would first consider a patient to be high risk, and then the information come back that they're actually not, that they were willing to go down in treatment. We're not advocating that one is better over the other, but what we do see is that there's a lot of opportunity to have that clinical discussion and to see what the optimal treatment path might be. The escalation principle is then also not referring to a single treatment. So you would also, in the conversations that you might have with your physician, see that escalation might be for a specific physician to do a tandem transplant or 2 stem cell transplantations. It could be to go from a doublet to a triplet to maybe even a quadruplet, to add more treatments to it. This really depends on the level of conversation and discussion you would have with your physician, to see if there will be a different consideration for the treatment path. And is it also something that you would feel comfortable with and are willing to choose for? Because as I believe it is your choice. And the same would apply to the escalation principles. What we then also see is there's not enough conclusive data out there to be advocating for a specific treatment. So this is where, as a company, we really try to work again with the physicians and the research groups, what might now be a specific treatment that might overcome that high risk. Until now, there has not been any publication out that demonstrates that that would be the preferred treatment to be advocating for this group. The other work that we're doing in the background, so this is very on the prognosis, is that we're striving to get more information on prediction. So I would say it's not the holy grail, but I mean, when it comes to precision medicine, everyone should strive for, can we find that specific biomarker that would tell you if the patient will be given this treatment, they will respond with these results. That would be the ultimate goal, because then you actually would know what then the specific treatment would be. We have some really preliminary data. This is again one of the abstracts presented at a conference, but as a research group and as a company, we're working towards getting more of that data and to working with others to achieve that. There is also more data out there that although you want something that would give you a positive prediction, so you want to have something that would give you, if you give this, the response will be well, we also have some data that, like I presented before, that if you are high risk on SkylineDx, you might not derive that statistical benefit, for example, on lenalidomide maintenance. Therefore, this can be a conversation that you can have, or that the physician might trigger. This is definitely a debate in Australia. So the physicians and the clinical guidelines there are really debating what this would mean for clinical practice. So if they know that the patient is high risk, would they still strive to give lenalidomide maintenance? Would they give something else, or would they not give it at all? And again, these are things that are still to be defined. But what it all plays into is that there's still a lot of room for improvement. And the exciting thing is that there's still a lot of new treatment options being introduced. We acknowledge and as a company fight for that unmet need and a better definition of high risk if it's newly diagnosed or if it's relapsed disease. But we also strive for information to correlate that to specific treatments. We do that with clinical research groups, but we also really try to collaborate where we can with pharmaceutical companies that are designing those trials to bring in that information of SkylineDx to then see if there actually is something that would do better because there's still a lot of improvement that we can achieve. This was my presentation. I hope you found it informative. I'll give it back to Audrey.
That was great. I found it super informative. Thank you so much for sharing. And I feel like you took a complicated topic and explained it in a clear way. Lots of questions. People are really excited to ask you their questions. One of the ones that I saw throughout was, how do I sign up for this Skyline? How can I get tested for Sky 92?
Yeah, so well, thank you for your question. So one of the things that we really wanted to do is do all the appropriate validations before we brought it to market. We found that that is imperative, that we really know that everything works well, that you get trustworthy results, and that the physicians also have some experience with it. So the test is available through the San Diego lab. So this is fairly recent, but we're now just bringing this to market in the US. We are, as we speak, working on a dossier to have a coverage discussion with Palmetto, MolDX, a Medicare administrative group, to also make sure that we have the appropriate evidence now translated to the needs of the payers so that there's coverage, but there is no coverage at this moment. So we're working diligently on that. And then the test would be available as of now for anyone who would like to get more information. We can make that happen. And I have to liaise with you to see how we need to do that, but it is feasible to do.
Yeah, and we can talk after this and just make sure that we have all of the right information. They get an email with this recording within 48 hours of the event ending. So you and I can collaborate information that they will need in order to know how to do that and how to stay updated on progress.
Yeah. Yeah, and one opportunity is always, sorry, Audrey, one opportunity would always be to reach out to the investigators that did the US trial. So if it's US specific, feel free to reach out to them. If you're not already familiar with these people, we can always facilitate an introduction by different means. But the website, like I said, is always in the recording, and you would see the investigators listed.
And remind us of that website one more time and the name of the trial.
The Trellis Promise, so it's PROMMIS, and the trial can be found on clinicaltrials.gov. If you type it in there in combination with either SkylineDx or myeloma, you would find more details on the design of the trial and the associated investigators.
Great. So we have someone saying Sky92 has been around a long time and GEP70 even longer. However, besides venetoclax for T1114 patients, do we have any therapies targeting any of these high-risk features?
Yeah, great question. Great question. So there are different initiatives looking at specific combinations to what I just said to overcome that high-risk profile. The venetoclax is really a targeted treatment. So the concept is there that if you have a specific mutation, then there would be a prediction of response on venetoclax, right? You would have a positive response to that treatment. We don't have that correlation for SkylineDx, but there have been different trials, and there's one ongoing in the UK that will present, I think ASCO, the American Society for Clinical Oncology, is next week. So there will be some early data present there. And we do see that more research is coming out, but there's not at this moment in time, unfortunately, a specific treatment that would give you a better outcome.
Okay, Thanks for letting us know. And I'm sure we'll see in years to come, hopefully soon, more and
Yeah,
more.
and the thing is as well, Audrey, is that for your audience is that the work that we do is retrospective, meaning that we look at past treatments because we need to follow up to really understand what is the progression. So there's always a time component in there that's unfavorable of doing the analysis. So you actually want to do it now, but then you have to wait before you can actually see if there's a positive correlation.
Interesting. That's really interesting. There are a couple questions about your slides. Do you have permission to share those slides with the participants?
I think so. I think the only thing that I wanted to check, I think I did, but just to be on the safe side, is that I've referenced everything accordingly and everything's correct. So I want to make sure that all the information that is there is retrievable online, and that people can access it accordingly.
Perfect. And I'll double check with you before we.
Yeah. Okay.
Do that. All right. So we have a question from Bonnie. She said, if a BMB is being used for MRD testing, NGS testing, and GEP, what poll do you need? Does it need to be a fresh slide?
Yeah, we need fresh bone marrow to do the analysis. Yeah.
Okay. When a patient is told that they are high risk, for example, 17p deletion, does that mean 100% of the multiple myeloma cells have this, or could it be only 10% of the cells?
Good questions. Yeah, it's a good question because okay, the different layers to the answer of that question. I'm gonna get different answers. One, when a doctor would say high risk, I think you need to ask the question, what does that mean? Make sure that you speak the same language and have the same interpretation. So what we see across the literature and all the discussions that we have is everyone has a little bit of slight understanding or different, they're differently convinced on what high risk is. Is that progression over a certain period of time? Is that not responding to a certain treatment? Is that, so the different aspects to that. The other thing is there are different methodologies to measure, for example, translocations. And then if you would look at deletion 70p, the methodology can differ between institutions, how it's done. But then they use a threshold or cutoff of how much is there to be giving the conclusion, yes or no. And that can be different. So if they would follow the guidelines, there will be a strict guideline on what it is. But it's also good to clarify that with your physician. But it's not that it will be communicated in, you have 10% deletion 70p. They will not give you that information, but they themselves for the analysis will use a cutoff for it to be communicated yes or no.
Okay. Okay. I think that clears things up a little bit. Thank you. Okay, and just a couple more questions of people asking, how can I have my facility send my specimen in for this test, and when it will be available in the clinic?
Well, you told me there's an international group of people potentially in the audience, so it's very different for Europe, as Europe has many different countries, many different challenges when it comes to the different countries. So that's very specific. But the US, a physician can reach out to our lab, or our colleagues in the US can help facilitate that, and we can make that happen.
Great, awesome. Okay, lots of questions are repeated. So if anybody has more questions that they want to enter, we have, we're getting some more. So and it's okay if you don't know the answers to these questions, by the way. For patients that carry the BRAF mutation, what is the current state of treatment with BRAF and MEK inhibitors?
This is also a good one. So BRAF-MEK combinations, it's a really well known combination for, actually, I said it wrong at the beginning of my presentation. for melanoma. So BRAF/MEK for melanoma, really well accepted and adopted. There is a trial in the US that's called MyDrug. The MyDrug trial looks at specific mutations. It's not in my deck, but I can provide that information if it's not already something that has been shared on your website. But MyDrug really looks at specific mutations like BRAF and then combine it with targeted treatments. And this is the benefit of the BRAF mutation, is there are treatments that immediately target that mutation in a specific principle and therefore work. They're not in label, if you will. So there is currently not a specific BRAF/MEK combination approved specifically for multiple myeloma. It does not mean that there's not a positive effect. And that trial looks at mutations like that with the ambition to then go into drug opportunities that might be much more known to other diseases or indicated for other diseases and have positive effects there, but can also be then correlated to a potential positive effect in multiple myeloma.
I think this brings up an interesting point, and I don't know if you can share with the audience. We're not only, as Skyline Diagnostics, you're not only working with multiple myeloma, but other diseases and cancers as well. So do you want to share a little bit about that?
Yeah, no, I definitely can. So as a company, we just work with those researchers often that did the discovery. So what you would see is the more data that's collected on patients, the more opportunity that you have to find common denominators, which can be referred to as a signature or a biomarker. That biomarker could tell you much more on disease aspects, the progression, the aggressiveness, or it can tell you something about metastatic propensity, the different attributes that you can find there. There's a lot published, but some are just really promising for patients to bring to clinical practice. But a researcher doesn't have that infrastructure. They might have the ambition, but they don't have the funds or the infrastructure to work on that. So when we found such a biomarker, we go through a due diligence process, which means that we would look at the robustness of the signature, the biomarker, the biology behind it. Can we translate this into a test? And then we would liaise with the institutes to then see, are you willing to work with us? This relatively small company in the Netherlands that has the ambition to bring tests to US patients or to European patients. That has led to a collaboration now 3.5 years ago with the Mayo Clinic for melanoma, so for skin cancer. And we have developed a test that on the basis of the primary tumor would be providing a much better understanding of the metastatic propensity. Therefore, if the patient should be considered to get adjuvant treatment, where they now might not get any treatment at all because they're kind of wrongfully so regarded as low risk for recurrence. So this is partly in the research phase, but we're doing that together with the Mayo Clinic. We're also working on something completely different, Kawasaki disease, which is more related to COVID-like symptoms, but it's an inflammatory disease in pediatrics.
Interesting. We're getting questions as well. Yours is called the Promise study. There's another Promise study by Dr. Gabriel. So let me just try to clarify the difference in my words, and if you want to add anything in there. So the Promise study by Dr. Irene Gabriel is for MGUS and smoldering patients and their families. If your two family members have had multiple myeloma or another blood cancer, or if you're African American. So that is testing. I can also send it in the follow-up email if that's interesting to people, but it is some completely different study, and it's spelled Promise, P-R-O-M-I-S-E study versus your guys' study, which is, how do you spell your guys' study?
Double M, no E. Then it's, it's Promise, yeah.
Yeah, it's kind of funny, I think, when those two studies are going on at the same time with relatively the name. I understand the confusion between the two.
Yeah, it is a good point though. Maybe to reiterate. So our test has really been developed for symptomatic multiple myeloma, so not for MGUS or smoldering. So we have not developed it for that specifically. It's developed for newly diagnosed or relapsed refractory multiple myeloma.
And then another thing here, people are wondering, a couple patients are wondering, we've talked about high risk myeloma, but do you think their precision diagnostics or precision treatments will change depending on if they have lambda only light chain myeloma? Do you know what that is? Are you familiar with that?
No, I am, but I'm thinking about if there is something I could say to specific treatments. And I think you should ask someone else to give you the more appropriate answer. Yeah.
I think it's interesting, right? Because kind of as you were saying at the beginning of this, myeloma is so complex and it's so unique to each individual. And
exactly.
even, genetic risks look different in different people. And so it's interesting to see all the different types and variations of multiple myeloma there really are. And how interesting it will be as this research continues to see what treatments we find that are specifically helpful in creating a dormant myeloma or MRD negative, hopefully myeloma in patients. So thank you so much for your time, Arjun. I really appreciate it. I really enjoyed your presentation, very informative, and I'm excited to share this information with our registrants through the follow-up email. I'll close with just a couple of announcements. All right, so our next meeting will be next month in July. We'll be discussing Know Your Myeloma Therapy with Darzalex FastPro. This will be a continuation of the Know Your Therapy series that we are doing within the Myeloma Crowd Community Program. We will keep you updated with the date and time as soon as we've coordinated with our key speaker. Summer months are always a little bit more difficult to find speakers, but we are in talks with somebody right now, so we hope to have that information out to you soon. You may be interested in other Myeloma Crowd community events we have coming up on the 8th. We have our SoCal Myeloma Community Chapter. That's at 6:30 p.m. Pacific. Dr. Amrita Krishnan, who is a multiple myeloma specialist, will be hosting an interactive conversation. On the 10th, we have 2 events. The first is our stem cell transplant chapter. Registered dietitian Katie Broswell will be talking to us about what diet we should follow after stem cell transplant, as well as sharing recent data that is perhaps different from what you've heard in your stem cell transplant evaluation. We'll also be having the myeloma caregiver chapter that evening. We're going to be interactively creating resources in order to help build our own care teams as caregivers. So recent studies have shown that a high percentage of caregivers end up with significantly worse health after they've finished their caregiving than when they began. And that is something that we want to avoid at all costs. So please invite your caregivers, your loved ones to this meeting so that we can make sure we're taking care of both patients and caregivers. On the 17th is our newly diagnosed myeloma patients chapter. We're going to be discussing why we need multiple myeloma specialists. Recent studies have shown that it can add up to 8 quality years of your life. So that's important to know. We're going to be discussing studies and also just through personal experience why having a multiple myeloma specialist is important. On Thursday, June 22nd at 1 p.m. is our nutrition and wellness. We're going to be talking about how to choose a satisfying snack instead of just stress eating, which I am completely guilty of. The link to sign up for any of those events and even more events I haven't mentioned today is located at the bottom of this slide and will be sent out to everybody, as I mentioned in that follow-up email, within 48 hours. Another thank you to our sponsors, Bristol Myers Squibb, Amgen Oncology, Karyopharm Therapeutics, AbbVie Adaptive Biotechnologies, Genentech, Oncopeptides, Decade Oncology, and though they're not on this list here, Janssen also is one of our sponsors that we're really grateful for. And thank you to you to helping us build a strong myeloma crowd community. It means so much to me that you took your time to be here, or if you're watching this recording, and we appreciate you and hope you have a great rest of your day. Thank you.